please make your way to the microphones right now. We've got about 20 minutes, so let's make the most of this opportunity. If you could keep your questions fairly brief and make sure there's a question mark at the end, that would be great. And what I'm probably going to do, as we settle in our chairs, take a seat, is I'll take two at a time. I'll just kick off very briefly with this idea of radical transparency in science. Now, that terrifies a lot of scientists because it also means that your science, there's an anxiety that it might be shut down before it has the potential to really grow wings. So is that an anxiety that you share in having this very public conversation about your work?
From my perspective, everyone I've talked to has said that they are more trusting of the research in knowing what is going on and being invited to have their say and make suggestions as to how it's different. Among scientists, most people seem to think it's a great idea. They just don't want it to start in their field.
Well, I think the challenge will be, what do you do when someone disagrees? There's something you want to do, and you think it's right, and you're very convinced, but somebody disagrees with you. How do you handle it?
I think that's a matter of civic governance. But then gene drive is always a matter of civic governance because it is a shared impact technology. So I'm not saying that we humans have great ways of overcoming disagreements like that, but at least the debate needs to happen in public, and then hopefully we can come to a decision. Maybe it will be wise.
I will be watching your experiment with great interest.
What if it shuts down the work? What if Nantucket and Martha's Vineyard, these communities that you're working with, or Maori communities that you meet with next week in New Zealand say, no?
Then we walk away. We have to walk away. And in fact, with the Maori, we have to walk away even if they don't give any reason that makes sense in terms of scientific potential consequences. Because the Mātauranga Maori traditional knowledge is adapted, it's evolved to that particular ecosystem. And so there are concerns that they might have, things that they might see that could go wrong, that they can see are wrong, but they're not going to be able to explain it in a way that we can understand that makes sense in terms of reductionist science, but I still expect it's probably a perfectly valid concern. So we're going to have to be willing to walk away if they say, no, this is inconsistent with our cultural knowledge.
So that's very interesting. Jennifer, have you shared some of the anxieties about this public dialogue that you've initiated beyond science and scientists?
How do you mean anxiety?
Well, an anxiety that the work, everyone goes straight to the embryo conversation. And in fact, there's a whole lot of other stuff that's interesting and not as risky and raises very different ethical questions before we get to the embryo and therapeutic end of things.
Yeah. Well, I think it's valuable to have that whole range of discussions, and the reason is that I think it's great to engage people in science. I mean, it's really exciting to see all of you here tonight, for example, because I think you're here because you're interested in hearing about this interesting science and thinking about what it means for the future. And each of us may have a different perspective on what we think is going to be how this science might affect our own lives, for example. But I think it's all of it plays into this, public interest that I think is so important to cultivate.
This is a really unique opportunity to ask a question of these incredible scientists. So please, thank you very much.
Hi, this is a question for Professor Doudna. Did I pronounce that correctly?
Doudna.
Doudna, pardon me.
Close.
So theoretically speaking, is it possible to correct severe genetic disorders such as albinism? And in doing so, would one need to synthesize melanin?
So I think for gene editing to have an impact in genetic disease, at least in the short term, we're looking at diseases that have a well-defined single genetic cause. And there's actually quite a large number of those in the human population, but then there's a much larger collection of diseases that have multigenic causes, and those will take, it'll be longer before we're able to do that. And in the case of albinism, it's something that would have to be. I personally don't know enough about the genetics of it to tell you a great answer to that, but just to say that I think in principle, being able to do that kind of genetic correction is clearly on the horizon. And what has to be grappled with is, for each disease, is it going to be possible to do that in an adult or a child, or would it be something that would have to be corrected at the level of the germline? And there's different sort of considerations for both kinds of therapies right now. But already there are clinical trials beginning for treatments that would be effective in people or children that already have a genetic disorder. It's amazing to me to think that we're already sort of entering an age when those trials will be ongoing. So it's something that is clearly coming.
And then there's an active discussion about what we define as a disorder amongst the disability activist community.
Yeah.
That might come up. Right there at the back on the right. Thanks.
Hi, I'm just wondering what you both think in regards to embryonic alterations. How much have you engaged with, or how concerned are you about things like unequal wealth distribution and the class issues, and how much you've engaged with other fields like philosophy, history, anthropology, sociology?
Thank you for that question. And do we have another one over here as well? Thanks.
Do you think gene drive will give power to organisms and ecosystems to give them tools to either adapt or overcome the consequences of climate change?
Well, on the first question about equity, that's such an important issue. I'm really glad that you brought it up. I think that, to me, when I think about the global impact of gene editing, I feel that it will happen first with agriculture, and I think that's something where, in principle, this could be a very equitable kind of thing. And already there are very active programs going on to develop ways of editing organisms, plants, animals that will be better adapted to certain niches to give better nutrition or to deal with drought and other conditions that cause difficulties raising crops, for example. I think that's one thing. It's also interesting to me that this technology, and you may have, hopefully you got this from both talks that you heard, is relatively easy to access. It's actually distributed by a nonprofit organization that will send research materials to anybody who requests them for just sort of the cost of the materials. So it's something that is quite a low barrier to get a hold of, and I think that means that it's going to be possible for scientists around the world, and this is already happening, to adapt it for their own purposes. And I think that's also something that's, it could go both ways. It could be good or bad, but I think it's, it has the potential at least to be very enabling to people globally, not just those in certain parts of the world. And then with respect to access to healthcare, this is a very active, ongoing discussion. Certainly in the US, we grapple with this issue right now of, healthcare insurance and thinking about who gets access to what kinds of healthcare. What do you do when you have a technology that can be used to treat someone, but the cost is $500,000 a patient? Who should pay for that? And what's the value, and how do we measure that? And so I think that the biomedical applications of gene editing are going to fall initially in that category, but of course one hopes that over time there will be ways to develop applications even for clinical medicine that will be widely useful for people and will not, where the costs will come down over time. But that's something that clearly has to be very actively addressed. It can't just be allowed to, sort of to be in the domain of companies, I would say.
Huge issue in America if you don't have access to health insurance to cover this sort of technology down the track. If it did become a clinical reality, then, you'll have the haves and the have-nots, and then, the have-nots will be discriminated against in all sorts of ways. Right. Huge issue. Gene drives and climate change and adaptation by organisms.
So this is a great question, and the challenge is what adaptations would actually be helpful. So we're not so great at creating any given phenotypic trait if we haven't already seen it before. In fact, with CRISPR, we can try it now. We can try a bunch of different combinations, but it doesn't mean we're necessarily able to adapt an organism. But if we have some examples of an organism that is succeeding in adapting, but it's we're going to lose most of the population before that adaptation can spread just because they're not getting selected for strongly enough. Then if it is only a couple of genetic changes, then yes, we could potentially use a gene drive, even in this case a self-propagating gene drive, potentially, to spread that adaptation rapidly through the remainder of the population and thereby rescue them. I haven't really looked at any specific cases where people have proposed doing this, and I'm not an ecologist myself, but I would be very surprised if cases like that don't come up.
What an interesting possibility. Thank you very much. I'll take your question and then I'll come to you for your question in a row. Thank you.
Okay, I'll be incredibly quick with you guys. Thank you for the international guests for coming. I think everyone here appreciates it. Just want a quick question for Jennifer and maybe just an idea for Kevin. Jennifer, regarding what you said about, the example you gave with the tomatoes and especially the example you gave with eugenics, creating babies with blue eyes and blonde hair and all these fantastic abilities. Do you think that, if you set that aside and then you've got a population of people who suffer from all sorts of cancers and, there's 7,000 orphan diseases out there, do you think that healthcare should take priority?
So healthcare should take priority over other uses of the technology?
Yes, over
All right.
eugenics, agriculture, etc.
Okay, thank you very much. And another question?
How will research on human embryos affect the respect we have for human life in her early stage of development? That is, if many human embryos will be destroyed in this research, do you think the end justifies the means?
Well, for the first question, I would say that my thinking there is that I think again, going back to a point I made earlier, I think that having the ability to do gene editing in plants has the potential to have huge health impacts on people, much more broadly than individually treating patients, if you're just thinking about numbers, right? And so I think that that's something that, at least in the shorter term, is going to have a larger impact, and I would myself prioritize that kind of work.
But the hype around that's been with us since the Green Revolution.
Yes.
That we would feed the world with genetic engineering techniques. Yep. We're not feeding the world.
No, but we have increasing challenges, I would argue, in that regard, right? We've got a growing population. We're facing the effects of climate change, and to have the ability now to make modifications to plants that don't require years of breeding, and to do that accurately, I think is, it opens the door to doing things that in the recent past were not possible. And I just want to make one other comment about that question, and that is that, I certainly think that we would want to, in terms of health, the clinical applications, that we would want to prioritize uses for diseases where there, were things that we would all agree are desirable to fix. So for people that have degenerative neurological disorders or that have diseases like cystic fibrosis or muscular dystrophy, these are diseases that are in principle treatable or potentially even curable using gene editing. And I think that's something that should clearly be prioritized. At the same time, we have to be grappling with the reality that when you have a gene editing technology like this, that it can be used for other things like enhancements and however you would define that. And so I think it's critical not to put that discussion on a shelf, but actually to be talking about that right now, because that technology is clearly right on the doorstep.
If I might interject on that one, I've sometimes been criticized for suggesting that we should think primarily about applications such as malaria for gene drive, and that we shouldn't necessarily risk taking a hit in terms of public support should something go wrong on something else. That is to say, the earliest applications of a new technology are critical, and we should absolutely prioritize those ones where everyone can agree that the problem exists and is severe. Because otherwise you run the risk of things turning out like people's suspicion of GMO foods. I mean, honestly, what have GMO foods done for anyone in this room? Like, yes, food is very slightly cheaper, but that's hardly something obvious. So I would say we should, whenever possible with a new technology, prioritize things that everyone can agree is an obvious and severe problem.
So this distinction between using CRISPR to treat patients who are adults and you might be able to then infuse them with gene-edited cells and so fix what's ailing them, versus do we start at the embryo stage and genotype an embryo, understand what risks are being conferred by its genetic makeup, and then edit at the embryo stage? Well, that's a question that's come up a lot for this audience in the questions that you sent earlier. So I think it also taps into this woman's question as well. Do we go anywhere near embryos?
Well, we're already going there. You know, people are already doing research on embryos. It didn't start with gene editing, though. You know, it started with, I would argue, with in vitro fertilization and all of the technology that went into that, and with something called pre-implantation genetic diagnosis, or PGD, which is now done routinely to screen human embryos for, and also animals, but humans, human embryos for genetic disease prior to implantation. And that's something that is now offered to people that are using in vitro fertilization quite routinely. And I'm old enough to remember back before there was in vitro fertilization, before and after, and I remember debates in my family growing up about, my parents saying, well, that seems wrong. Why should people be, making babies in a test tube and test tube babies? And there was lots of discussion about that, and it seemed really wrong to certain people. And then over time, when it appeared that, Louise Brown was born and she appeared normal, and other parents who were otherwise infertile were able to help to have children, people over time came to accept it. And I think now, at least in many parts of the world, that technology is widely accepted. Will the same thing happen with modification of the human germline using gene editing? I don't know, but I think we will see evolution of people's thinking about this as the technology advances. And I would also point out that I think that in the not distant future, I can't tell you exactly when, because it's very much on the sort of the bleeding edge of technology right now, but I think at some point it will become possible to do the editing in germ cells. So that means in sperm or eggs rather than in embryos. And that sort of removes this whole, I think, for many people, ethically difficult issue of, do you really want to be working with embryos that then have to be discarded? It feels uncomfortable in many ways. And so I think if we could be doing editing in germ cells, the sperm or eggs, and then selecting those cells that have the correct desired edits, and then using those for fertilization, then I think that's potentially a more appealing way of using germline editing in the future. And we're not, like I said, we're not there today, but I do think the technology is moving very steadily towards that moment.
And we should point out that most of the work that's being done on embryos, they're non-viable embryos, so they will never be implanted, they will never become a baby as such. But what that's allowing is scientists like those at the Crick Institute who have just published to understand the development of what genes are involved in development. And that's vital, isn't it, for understanding humans and our biology.
It is, yeah.
Did you want to add anything on that, Kevin?
Well, I'd just like to say that I don't touch human editing stuff because I don't like doing anything controversial.
There you go. No, nothing controversial, my foot. Okay, so could we go to the back and then over to this side? So that one over there.
Thank you. So in order to make GeneDrive a pioneering example in open science, I think you have to do more than just make the information and the experiments and the suggestions available. You actually have to make people want to read them. So how can we make the shift to a more scientifically literate society that's actually engaged in these things, and how long do you think that will take?
Okay, and thank you.
Thank you both for your work and your talks. It's really great to be here. Just addressing two of the concerns that have come up tonight around human gene editing, genetic haves and have-nots, and also just concerns about changing human nature. There's another biological technology vaccinations that initially were only available to developed, wealthy countries. Do you think in the future thinking will evolve and it will, like nobody now thinks that vaccinations are a bad idea, well, nobody scientifically literate thinks it's a bad idea, and they save countless lives. Are we going to go in the same direction, or are there important differences between these two technologies?
I'm not going to repeat those questions. So the first one, though, was looking if we want to engage with open science and make gene drives the case study, how do we better engage the wider community in science and scientific understanding?
Well, I think Convergence Science Network and events like this one are certainly helping. But I think a critical aspect is inviting people to submit suggestions and share their concerns and then actually acting on them. That is, we shouldn't just tell people about what is going on as though they're passive recipients. We need to actually invite them to help shape the future because they will share in it. And I've found that we've managed to involve many, many community members who said that they were generally skeptical of science or wouldn't normally have considered this sort of thing, but if it's going to be done in the open and it's going to invite concerns from everyone and that their voice is going to be listened to, then they're very glad to participate. And I'm very glad to have them because, frankly, the people I really want joining an effort are the ones who are the most skeptical of it because they're the ones who have every reason to find something potentially wrong with it.
I'll come back to that. I've got another question about that. The vaccination question, using that as a sort of platform for what's to come.
Yeah, well, I think that's an interesting example because, you know, I live very close to Marin County, California, which is a county where there's a lot of highly educated people who reject vaccination. Even now, they will not vaccinate their kids, and we've seen outbreaks of measles and other things like that that result. And I think it speaks to the issue. It kind of actually relates to that first question of, you know, how do you engage people in science so that they understand enough about technologies that are coming along that are affecting our lives so that they can make responsible and informed decisions about using those technologies. I also think that people, people's views can change over time. You know, if they're open to discussion and to looking at sort of facts and data and thinking about it, their views can change. And back to the in vitro fertilization example. That's an example where people, I think, initially who were skeptical or felt that this technology was not something that they would want to use, when they saw that it was safe and that it had a very positive impact on a number of people's lives, maybe even their own lives or someone in their family, that for them then became a transformative experience. I'll tell you a very brief little story. I went at one point to Congress and in the United States, and I was discussing this technology and what it would mean for the future. And I found myself sitting across from a very conservative rookie first-term congresswoman from the Midwest who was known for her extremely socially conservative points of view. She was someone who really did not support any of the sort of new technologies and was kind of known as a somewhat science skeptic, I guess you could say. And what was fascinating, I was wondering how this conversation would go between her and somebody like me, and it turned out that she had given birth to a child the year before, that while it was in utero, while she was pregnant, they discovered the child was missing a kidney. And this required them to ultimately do fetal surgery. And it was a very difficult thing. Fortunately, it had a positive outcome for her and the child. But she told me that, she said, if the CRISPR technology was available at that time for my child, I would not hesitate to use it. So for her, it had become a very personal thing. And so I think that it's really important that we find ways to engage with people on a level that is relatable to their lives. And I think like what you heard from Kevin, I think is a very good point as well, which is that we need to ensure that some of the earliest uses of new technologies, and certainly for gene editing this is true, that we are collectively selecting applications that are going to be positive. They're going to be viewed as things that are solving real problems, not just making money for companies and things, but really actually tackling problems that people are facing in their lives. And I think if we do that as scientists, that will go a long way towards engendering goodwill and the willingness of people to engage with us and think together about how we go forward with this in the future.
But yeah, we could talk for hours about the commercial imperative versus open science,
I do have one brief anecdote to add,
Yeah.
and that's just that for Mice Against Ticks, there are some people that we've met on the islands who don't vaccinate their children and would never knowingly eat GM foods. And yet they're wholehearted supporters of the idea of releasing 100,000 genetically engineered mice on their island.
because they don't want Lyme disease.
Because they don't want to get Lyme disease.
Lyme disease.
So-
I'm going to take one more question, and that looks remarkably like Ellen Trounston, but I might not be right about that. Is it Ellen Trounston? So that's Ellen Trounston, who trailblazer in IVF.
So Jennifer, great to see you. It is really an incredible technology, and I have every confidence that we can use this really wisely for the benefit for all kind. And I have absolute confidence we can do that. Gene driving, that's a different gig, I think. And so I like the idea of it being open. I think that's a great idea. But somehow we have to figure out how to fund all that. But what I wanted to ask you, Kevin, was isn't there ways of sort of maybe introducing an antisense to recover back the population if it went wrong, or insisting on putting in a suicide gene of some kind that would sort of, collapse that population if it was doing something that was unpredictable? And couldn't we wind those kind of things in that we do in some medicines, to make sure, well, we'll get rid of this if it's not doing good? Can't we sort of build those things into it? Because you're right, it does worry me that once it's gone and out of control, you can't stop it, and it only stops when it's fully complete. But you must be able to back it off in some way.
Yeah, are there other ways other than daisy drives, to pull it back once it's out there?
So there's a lot of research going into this, funded by a number of different organizations. And the challenge is to make it evolutionarily stable. Because you're right, including a inducible toxin or something like that, trying to include a kill switch, but then you run the question of what triggers the kill switch. Is it an environmental chemical? Can you be sure you get all of them? How is that different from just using an insecticide to kill all of them, et cetera? But the thing about gene drive that really makes me quite relieved, and in fact, the first thing we tested in yeast was, of course, can you overwrite an earlier gene drive with a newer one and restore the trait that was changed by the first one? And the answer seems to be yes. And thanks to the versatility of CRISPR, because it can target so many different sequences and there's so many different CRISPR enzymes that have been isolated, it's impossible to make a sequence that is functional and yet can't be targeted with CRISPR. Meaning, no matter what anybody does with a gene drive, we can always build another gene drive to undo it. And if we can figure out something that is sufficiently localized, whether DAISY or something else, we actually have a project that builds what we call DAISY threshold drives that would be even more tightly controlled and would essentially link it to a genetic change that makes the genes go, causes natural selection to act against them if they're in the minority. With the idea being that then we can use this to alter the ones that have been changed in undesirable ways, and then release a suppression drive specific to that particular change that would reduce the population relative to wild type. Once they were in the minority, natural selection would clean them up, and you'd remove all engineered genes from the population. And whether or not we need that, I'm actually skeptical in most cases of potential environmental damage. The reality is that many people feel that nature is sacred to them. And the notion that we would have introduced engineered genes at all bothers them on a profoundly moral level. And I think we're obligated to respect that, even though I don't feel that way about it. I do feel compelled to try to develop tools that would allow us to remove all engineered genes and restore populations to the way they were initially. The thing we need to be careful of is we have never before engineered something that we anticipate evolving on its own. And having worked a lot with microbial-directed evolution and the like. I'm skeptical of things like kill switches just because it only takes one to escape, right? So we do need to be very careful, and even with something like Daisy Drive, the reason we're working with testing it in worms is because evolution is a numbers game. We need to be able to grow hundreds of millions or billions of the relevant organisms and see how stable is it, what is the escape rate. And so we need organisms like worms to do that. And then the question is, how much is a worm like a mosquito, or a rat?
Or a cane toad.
Or a cane toad.
Or a cane toad. Thank you very much, and thank you for your questions. Sorry we couldn't get to them all, but please spill it onto Twitter and use that hashtag, and maybe the conversation can continue. And Kevin, I know, is active on Twitter, and he might happily engage in conversation. Sorry to put that to you. I want to acknowledge the sponsors of tonight's event. 2,000 of you registered to be here, and this is not possible without these amazing people. The Club Melbourne Ambassador Program, the Victorian Department of Education and Training, the Monash Biomedicine Discovery Institute, the Graham Clark Institute, the Murdoch Children's Research Institute, Cartherix Pty Ltd, the Faculty of Science at the University of Melbourne, the Australian Regenerative Medicine Institute. These are all Melbourne institutions. The Hudson Institute of Medical Research, and of course, your organisers of today, the Convergence Science Network, Luan and his incredible team. They would also like to acknowledge the support of the Lawn Genome Conference, who brought Jennifer out, and the School of Biosciences at the University of Melbourne, who brought Kevin out. And thank you to them for their generosity for making them available for this event too. So please thank Jennifer Doudna and Kevin Esvelt. Thank you.